A research vessel, Steel Hull Groening, pushes through the brash ice of the Southern Ocean. Today is January 29th, 2026. On board, a small team of virologists prepares for another day at the bottom of the world. They are here for a singular, profound purpose: to search for life in a place that seems defined by its absence. Their mission is not one of dramatic conquest, but of patient observation. They seek viruses, the most abundant biological entities on Earth, hidden deep within the Antarctic ice sheet. These scientists are ghost hunters, searching for the spectral signatures of life that have been locked away for millennia, far from the sunlit world we know. Their days are a rhythm of cold and concentration. Outside, brutal minimalism; inside, meticulous detail. Antarctica presents itself as a desolate, sterile continent. From a distance, it is a vast desert of ice and snow, seemingly hostile to all but the hardiest life forms. But this perception is deceiving. Life here has not been vanquished. It has simply retreated into smaller, more protected spaces. Within the ice itself, in the frigid waters beneath it, a complex microbial world flourishes. These are communities of extremophiles, organisms exquisitely adapted to the cold. The key to this hidden life is water. Even far below freezing, salts and pressure carve pockets and channels of liquid brine. Here, single-celled algae perform photosynthesis. Bacteria, archaea, and algae eke out a living. The process of finding new viruses begins with the deceptively simple act of collecting ice. The team selects their sampling sites with immense care, guided by satellite imagery and geological surveys. They look for areas where ancient ice is accessible, the face of a glacier, deep within an ice shelf. Promising samples come from long ice cores, time capsules trapping microbes and viruses. Other samples are taken from sea ice, with its unique microbial communities. Fieldwork is extreme precision. Contamination is the enemy. They don sterile suits, face masks, and multiple layers of gloves. All tools are meticulously sterilized: drills, saws, collection containers. The outer layer is shaved away to access the pristine interior. Each sample is sealed, and details logged in waterproof notes and a digital database. Location, depth, time, temperature, metadata as vital as the sample itself. Back inside the relative warmth of the mobile laboratory, the next phase begins. The meltwater now holds suspended microbes and viral particles. First, they separate components via filtration and centrifugation. Filters with progressively smaller pores capture bacteria, algae, and finally virus-sized particles. Tangential flow filtration concentrates viral particles into a precious small volume. Centrifuges pellet viral particles while debris stays in solution. They extract DNA or RNA by lysing protein coats under biosafety protection. The nucleic acids are fragile and scarce. Every step is exacting. The genetic material is prepared for sequencing, where the invisible becomes visible. Fragments feed into a portable sequencer that reads genetic letters, the A's, C's, G's, and T's. The machine yields millions of short reads, a snapshot of the viral community within the ice. The sequencing machine produces a torrent of raw data, millions of short fragments. Using powerful bioinformatics, the team assembles short reads into longer contigs, reconstructing partial or even complete viral genomes. Assembled sequences are compared against global virus databases. Some align with known families, bacteriophages or algal viruses, cold-adapted variants. But the most exciting moments come when no significant match appears. These novel sequences point to lineages evolving in icy isolation for eons. Finding such a sequence is like discovering a new species. The team annotates genomes, predicting genes and proteins. Hallmarks like capsid and replication enzymes confirm viral origin. Each novel genome is a new book, its unique code the signature of an unseen form of life. A single novel sequence is not enough. The core principle here is proof, not hype. They analyze controls, sterile water, equipment swabs, to rule out contamination. If the sequence appears only in the ice sample, confidence grows. They repeat the entire process: thawing, filtration, extraction, sequencing. They verify with different platforms and reagents to avoid method artifacts. Brick by brick, they build a robust case and share data and methods openly. Subsamples go to independent labs for confirmation. Sequences are uploaded to public repositories for global scrutiny. Publications emphasize methods, statistics, and limitations. A sequence alone cannot confirm activity. Only careful assays can. Proof over hype. Discovering a new genome is one thing, understanding its role is another. To learn function, they must identify the host. They build a catalog of potential targets from the original community. Some viruses integrate into host genomes. Matching fragments can be a smoking gun. Host-like genes inside a viral genome offer strong clues. These links map an intricate ecological network in the ice. Some viruses prey on abundant bacteria. Others specialize on algae at the food web base. When viruses burst cells, they release carbon, nitrogen, and phosphorus back into brine, a viral shunt that recycles nutrients and sustains life. Across depths and sites, viral communities shift with hosts and environment. Viruses are active shapers, deciding which microbes thrive or perish, sculpting biodiversity. The search for unknown viruses carries a profound sense of responsibility. They operate under strict safety and ethical guidelines to protect people and the Antarctic environment. The Antarctic Treaty designates the continent for peace and science. Activities minimize impact. Waste is contained and removed. Biosafety protocols are stringent. Human risk is considered extremely low, but caution prevails. Any culture attempts occur in BSL-2 or higher labs with controlled airflow and decontamination. Ethics reviews, responsible sharing of samples, open methods and data build trust and accelerate discovery. They avoid speculation, focusing on evidence, reliable witnesses to a hidden world. Safety and ethics guide every step of exploration. The discovery of novel viruses under the Antarctic ice opens a new window into our planet. It confirms a complex virosphere in one of Earth's most extreme environments. New sequences prove there are unexplored branches and adaptive strategies we barely understand. Yet the samples cover only a tiny fraction. Are these viruses widespread or local? We do not yet know the full diversity here. Are they actively replicating or dormant, awaiting change? New in situ methods are needed. As climate warms, melting may release ancient microbes and viruses into the Southern Ocean. What happens when they meet modern communities? We do not know. They may fade without hosts or subtly alter the marine food web. These are questions for careful, continued study. Facts, not drama. Sampling, sequencing, verification, each new genome a small piece of a grand puzzle.